So now it is my great pleasure to introduce today's presenter. Dr. Mark Lescroart is a postdoctoral researcher in the Gallant laboratory here at UC Berkeley. Mark got his PhD in 2011 at University of Southern California, working with Irving Biederman. Mark also went to USC for his undergraduate degree, graduating summa cum laude in 2002 with a Bachelor of Science in Psychobiology and a minor in Japanese. Mark has also written popular scientific articles for Scientific American Mind. So many of us believe we recognize objects in our world as a function of seeing through our eyes, but there's more to it than that. And in today's talk, Mark will help us understand the way our brains transform patterns of light in our retinas into useful information about the shape and structure of objects in the world. So enough from me. Please join me in welcoming Mark Lescroart.
Thank you, Rachel. Great to see so many people interested in vision in the brain on a Saturday morning. There's a better way to spend it. So I'd like to talk to you today about mind reading. But I'm not going to talk to you about mind reading because that's a bit of a fraught topic for a couple of reasons. First is that there's a great deal of debate about what the mind is and whether the things that our lab does actually constitute mind reading. Some people think that in order to read a mind, you actually have to get at the intentions or desires of the person whose mind you are presuming to read. And that's not what we do, for a couple of reasons, mainly because our lab is of the opinion that it's not quite time to do that yet. Not only because of ethical issues, but because the technology just isn't there. So we stick with what we think is the most stable way to get at the contents of your perceptual awareness, which is to study vision and perception. So I'm going to be talking to you mostly about vision, and I'm going to use an attempt to read out the contents of your perception as a way to teach you a little bit about the visual system. I would like to start with the other reason that I'm not going to talk to you about mind reading, which is how many bad bits of mind reading there are out there. Have any of you seen the show Fringe? This show made me cry on its very first episode, because in the first episode there was a scene in which there was a man who was unconscious and they had to decipher something, or they had to find out something from him, but he was asleep, he was in a coma, and so they had to somehow read something out from his mind. And to do this, they decided to establish a connection between minds, which is actually much, much harder than just reading out a single mind, but never mind that. The way they went about reading the mind was to use a single wire to measure all 120 billion neurons in the brain. And they aimed for the spinal cord, which is a great place to get at the brain, being that it is not actually the brain. They used more electrodes to measure their subjects' breathing and heart rate than they used on the brain. They situated the subject in a vat of watery goo for some reason. I'm pretty sure that was just so they could get her in something like a swimsuit. They watched their measurements of brain activity evolve in real time on this very complicated display, and they made sure to keep a hard copy on paper, which is always the way to do modern science. And they interpreted all this by eye, without any statistics or any kind of mathematics, just watching these traces go by. And there was a particular scene in which the woman who was making contact with the unconscious man shared a bit of a romantic moment with the man, because they were actually lovers in the story. And as this happens in the dream sequence that is unfolding, you watch the little squiggles do this. And the man who is the scientist in charge of this sort of nods knowingly. And it made me cry, because really that's just an insult to the complexity of the human spirit, to think that something as complex as your emotional state and your perceptual state, and all of that can be summed up by these 4 squiggly lines. At the very least, you need some more lines. So my favorite take on mind reading actually comes from Harry Potter. And there is a scene in the 5th or 6th book when Harry is learning occlumency from Professor Snape. And if you're not familiar with Harry Potter, Professor Snape is something of a nemesis to Harry, but he's helping Harry to defend himself from mental intrusions by Lord Voldemort, the evil bad guy for all of the books. And Harry's recently found out that Lord Voldemort can somehow access some of his thoughts. And Harry says, "Do you mean he can read minds?" And Snape says with this delicious derision in his voice, "You have no subtlety, Potter. You do not understand fine distinctions. Only muggles talk of mind reading. The mind is not a book to be opened at will and examined at leisure. Thoughts are not etched on the inside of skulls to be perused by any invader. The mind is a complex and many-layered thing, Potter. Or at least most minds are." So if you want to get at the brain, the things that you want are precise measurements in time and in space. You want measurements that are directly related to neural activity, and the more measurements you can get, the better. So there are a few ways to measure the workings of a mind, many of which actually do involve dropping wires into a living, breathing, working cerebral cortex. This is difficult to do in humans, mostly because of the skull, and because the spinal cord is not actually an acceptable place to record. Not if you want to understand the workings of the mind, anyway. So we use fMRI, which is functional magnetic resonance imaging. This meets some of these criteria in that it is reasonably precise in space. You can measure a small bit of the brain that is about 2 by 2 by 2 millimeters, about the size of a lentil. And you can measure these little sort of lentil-sized cubes across the entire brain. So you get a lot of measurements, on the order of 30,000 across the entire cerebral cortex. Unfortunately, these are not directly related to neural activity. They are related, but not as directly related as many people would like. So to directly measure neural activity, what you want is a measure of electricity. And this is fundamentally a measure of blood flow. So my advisor, who likes to say that he has done real neuroscience and measured from actual neurons, likes to compare this to trying to guess the population of a building by counting the number of toilet flushes, which will be related for sure, but not as directly related as you would like. Also, these measurements take 2 seconds to collect for the entire brain, which means that you get one sample of brain activity every 2 seconds. And this is not bad, but this is not so good either. Neural events unfold very, very quickly. So what we're reduced to is understanding what's going on over a wide swath of the brain in a fairly coarse time increment. So there's been sort of a fashion lately to hate on fMRI. I don't know how many of you read science blogs, but there are a lot of people that call various fMRI findings neurobunk, neurobollocks, or neuroflapdoodle was my personal favorite. Are there any out there that like to hate on fMRI? Unfortunately, it's the best we've got. So this is what we're working with. So I'd like to get that out of the way up front, just to remind you that we're not actually measuring neurons, and that you should take all of this with a grain of salt. And if you ever want to ask me, wait, how do you know that? Can you explain a little more? Those are my favorite questions. So please do. And I may have to hand wave just because we've only got an hour and some of this stuff is a little complex. But anyway. So an MRI scan, like I said, gets you a grid like this across the brain. Each measurement is about 2 by 2 by 2 millimeters, which is going to be hundreds of thousands of individual nerve cells within each one. But hopefully they're all doing something similar within this sort of tightly localized region of the cortex. And for each one, we can get some activity measurement for each point in time. And for a different one, we get a different activity measurement. For a different one, we get another activity measurement, and so on, up to a great many more. Yes?
What limits the resolution?
There are a number of things that limit the resolution, including the fact that this has to be done on a human brain. And you can't zap them that strongly. There are also mechanical limitations for how fast. This is actually measuring the get this exactly right, the magnetism of a particular very localized chunk of the brain. And the thing that most strongly affects it is hemoglobin, which is a ferromagnetic compound in your blood. So the way that you are able to spatially localize different places is to set up a magnetic gradient across the person's head. That's what this great big tube is for. It's a very, very strong magnetic gradients inside. And so the gradients have to change to be able to measure different locations, and there are physical limitations on how fast those gradients can switch. And there are physical limitations on how much energy you can then zap into the person's head to then read back as it resonates out. Good question. So you get a bunch of measurements, and what this amounts to, essentially, is a code, a barcode, for some perceptual event. And different perceptual events with different stimuli, or different states of attention, or different degrees of wakefulness will have different codes. And our job as neuroscientists is to interpret each of these codes and see what we can make out of this brain state and say what was happening in this person's brain right here. And the best way to do that, the only way to do that, is to consider each individual bar on that bar graph. So to take one measurement and measure it over time in response to some stimulus. So we stick people in MRI scanners and we play movies for them. And we get measurements like this at 30,000 different locations across the surface of the brain. And our job is then to say, why is the signal high here, and low here, and high here, and low here? What happened that was particularly exciting at that location? And remember, different locations in the brain will be excited at different times. So there are different functions that different parts of the brain perform. So since we're talking about vision, mainly what the visual system can be thought of as doing is filtering the input that you get from your eyes. Any photographers out there? A few, okay. How many of you use filters in your photography? Fancy photographers? A few. So if you put a red filter on your lens, that means that anything that is blue or green or yellow is going to be reduced, and only the red is going to get through. So this is more or less what each individual neuron in the visual system is doing. It's taking all this light that is bouncing off of things in the world and coming through my pupil onto my retina and responding to only some part of all that information. Partly this is because light projects to a very particular spot on your retina, so one cell on your retina can only be responsive to something that is a very particular direction from your eye. But on in the visual system, each stage of processing, where some neurons connect to some neurons, those neurons connect to some more neurons, this is a very good analogy for what's happening, that the input is being filtered. It's being, some information just doesn't go through and some does. And there are different kinds of filters that different parts of the visual system can be described as implementing. For example, there could be a color filter that would pass only red stimuli. Or there could be some kind of a location filter that only responds to a very particular spot in your entire visual world. We call the visual world the visual field in the study of vision. And that basically is just everything that you can see. And it's measured in degrees from 0 over here to, or negative 90 over here to positive 90. And it's not actually 90, it's more like 75 or 80 degrees in each direction. So each neuron responds to some window onto that world. And I'm going to show you an experiment that very clearly demonstrates that. So the simplest kind of experiment you can do to figure out what the brain is doing is to show very simple patterns like this that are just contrast reversing checkerboards that then either rotate around the screen in a clockwise or counterclockwise direction, or that expand, so this ring basically shrinks or grows depending on which instance of the experiment you're running. And what I'm going to show you now is neural activity in response to those stimuli. Okay? So this is a brain. I know it doesn't look very much like a brain, but maybe this will help. Okay? So this is a brain, and the part that's white is the part that we actually scan. We don't always scan all of the brain. We try to most times. But for this particular scan, we're really only interested in the back, which is where the vision happens. So let's see if I can give you a bit of a marker here. So we're looking at this part of the brain, all the way toward the back at the occipital pole. Okay? And what I'm going to show you now is the activity in response to a ring stimulus that was slowly expanding. You're going to see what happens with the brain activity.
We're looking at the occipital lobe here.
This is the occipital lobe where I put this marker right there, yes. If you want to see it one more time, that's the back, that's the front. It's a little dark, sorry. So, what happens is that the activity looks very much like an expanding ring itself. As if there is a map in the back of your head that corresponds very precisely to the map of your retina. But the topology of your cortex is the same as the topology of your retina, and so we say that the visual cortex is retinotopic. And it's not just in this dimension of how close you are to the center of gaze. It also holds for rotation. So if we have a clockwise wedge that starts in the upper right visual field, so it starts up here, you can see that most of the activity is down here, in basically 1/4 of these areas that are sort of marked out in the brain. This is pretty cool. This then, as the wedge rotates, the wedge is now rotating clockwise to the right, so it's going to now move to the bottom right of the visual field. And the activity then shifts to a different quadrant of the brain. Yes?
So I'm sure you're familiar with these experiments that they've done where they take glasses and people, and they flip their view of the world, and eventually after a couple of weeks or something, they learn to see. They're normally seeing things, even though whatever it used to be from the top is now coming from the bottom and vice versa. Do you know what that does to this map on the back of your brain?
Well, this map stays constant. What changes is actually the readout function. So what changes is how the more advanced stages of processing in your brain interpret what's going on at these stages. So these stages are actually quite straightforward. As you can see, I mean, you show something and you get a very reliable response just based on the location of the stimulus. The full story is going to be a little bit more complex than that, but these are very basic perceptual areas. They're very tightly locked to what's going on in the outside world. Their job is to tell you what's out there.
So I guess one brief question, this is very important to me. If you have a retinal, like a retinal detachment, retinal surgery, and your retina moves slightly before it's replaced, from what you're saying, I would guess that then what you see would move in the back of your brain. Is that your understanding?
That probably would not happen. If that did happen, that would be very bad. But for that to happen, what would have to happen is that the connections from your retina to the rest of your brain would have to be severed and reattached. And I'm pretty sure that does not happen.
No. All that happens in this case is your retina, let's just say it's rotating slightly in your eye. And that would be equivalent to rotating, having glasses that would rotate the image.
It would be exactly equivalent to that. And my guess is that the same thing would happen. That now you just have light projecting in a different way into the back of your eye, but your system can handle it. So the map on the back of your brain would look maybe somewhat strange from the perspective of the screen in front of you. But from the perspective of your retina and where those cells grew up, so to speak, it would be constant. So let me finish our little rotation here. By the way, that question was something that perplexed the first scientists that studied optics for a long time, because the image on your retina actually is upside down. Things that are above you pass through the hole of your pupil and onto the bottom of your retina. And people thought, this can't be true. The world doesn't look upside down. How could this possibly be the right model of the way that light moves if it means that the world is upside down inside our heads? But it turns out the answer is, it doesn't matter. It's fine. So the activity we're following started at the top right, top right, moved to the bottom right, this way. Now it's going to move to the bottom left, which is going to be another shift, looky there, and then it's going to shift back to the bottom right. Okay? So this means, let me roll this up for you just a little bit and show you a very nice aspect of this. So this stretch from right here to right here is what's known as primary visual cortex. And you can we determined that it is a single area because it has a full representation of half of the visual world. You can see that activity as the wedge rotates basically passes right across it in a very smooth progression from the top down to the bottom. From the lower vertical meridian to the upper vertical meridian as that wedge circles around. Okay? So most of the first part of this talk is going to center on things that happen in this primary part of visual cortex. Yes?
The image you get into your optical nerve, how does that physically move from your eyeball to this area?
Great question. So there are back pocket is about 1 million neurons that are the last layer of processing in your retina that then go out the back of your eye into a structure in the middle of your brain called the thalamus, the lateral geniculate nucleus in the thalamus. And the way that those fibers are all gathered and project is one neuron projects from a spot on your retina out several centimeters out to the thalamus. But in all that distance, all those fibers are kept in exactly the same arrangement, such that when they form synapses and connect to cells in this structure in the middle of your brain, the organization is preserved. So it's basically just this incredible axon guidance that keeps things from getting completely scrambled. And that is an interesting question for how that develops, but that is by no means my specialty. I just study the finished product. So what this means is that now we have a way to start interpreting these signals. I told you our goal is to decide why the signal goes high and why it goes low. And so now we have learned something. We didn't actually learn this through fMRI. We learned it through other means. But we've found very beautiful maps of those smooth progressions through visual cortex like I showed you using fMRI. But now we know that cells somewhere in the back of the brain have a particular window onto the world, which is this little circle that I've drawn over there on the retinotopy stimulus. So as the wedge rotates, we know that the activity is going to go up when there's something happening, some contrast reversing checkerboard pattern that comes over here. And that's probably what happened there, and there, and there. And so basically the points where this was low would be something that was just outside that little window onto the world. And we call those windows onto the world the receptive fields of neurons, or by extension of voxels. I'm sorry, I'm not sure if I told you this, but a voxel is a volumetric pixel. Simple word. Hopefully you can remember it in one shot because I'm definitely going to say it again. But each of these little cubes is one voxel of the MRI scan.
Spell it. Spell it.
V-O-X-E-L. So now we have some insight into what's happening. And so we can begin to try and decode what's happening in the brain because we know something about, well, if there's activity, there must have been something here. So an interesting corollary of this is that if you have a head injury, get whacked in the back of the head or even the front of the head and your brain sloshes into the back of your head, and you damage part of your visual cortex, you get a blind spot. You get a scotoma. Because it doesn't matter that all the light is still getting into your eye and your eyeball is just fine. Both retinas are totally intact. Once those connections reach this particular spot in the primary visual cortex, they project to the thalamus, and from there they project back to the back of the head. The neurons there are dead. And the message doesn't get passed from there on. And so this manifests itself as just a fuzzy spot out here. Anyone know anyone that has had this happen?
Well,
Yes,
I
question?
had an experience where a concussion hit me in the back of the head, and I got to visualize an explosion in slow motion. So what is the mechanism that would cause that?
Do you remember that Far Side comic with the neurosurgeons pressing on the guy's brain and foot kicked out?
Yeah.
So that was a good one, Frank, try this. So if you did that in the visual cortex, what you're describing would happen. If you press on the whole visual cortex. If you were to stimulate more precisely within, say, one of these voxels, what you would get is a phosphene, which is a flash of light that's very localized in space to wherever the receptive field of that neuron is.
Yeah, but how does that whole vision slow down? Because I was actually seeing the explosion and realizing I was getting blown up. I actually saw the explosion, the billowing dust in slow motion coming on in and being able to realize that I am being blown up.
Does anyone mind taking a little derail on this question? So that's a very interesting question. This is a commonly described phenomenon that is a perceptual slowing down around traumatic events.
What kind of phenomenon?
It's just a perceptual slowing down.
Perceptual.
So the question is your visual system actually working double hard? And is it like double sampling the world in that time? Or is there just something about the visual input that just gets more strongly stored in memory, maybe to the exclusion of other things?
So how can you slow that time down?
Well,
you actually see it.
Maybe your neurons just work a little bit harder. Maybe they fire, activate, maybe they fire electricity at twice the rate they usually do.
Well, is there some chemistry that is created there? Or is it just like, does chemistry create the thought,
So
the thought create the chemistry?
The only experiment, well, the answer is they don't double sample. That it must be something that you just remember that so much the better. because of other mechanisms that are outside of the visual cortex.
Okay,
And this was
so
tested.
There could be another mechanism like the final body releasing DMT or?
Sure.
Okay.
Yes.
So it is the chemistry.
Yes. Well, but the point is it's not a double sampling of the world. And the experiment that speaks to this, sorry, this is a bit of a derail, but it's lots of fun, was a scientist at Baylor named David Eagleman, who writes a lot of pop science stuff. You might have read some of his stuff. He did an experiment to test exactly this question, which is, does time slow down, or do you double sample the world when you're stressed, when you're extremely afraid? So to make people extremely afraid, well, first to figure out how fast you're sampling the world, he had a wristwatch that had 2 digits, like a 6 and an 8, that would alternate back and forth very, very quickly. And they would find a speed at which they were alternating just faster than the person could make out what the 2 digits were. And so if you actually do double sample the world, then you should be able to look at that and see what the 2 digits are. So he straps these wristwatches on people's wrists, and then he brings them to the top of a roller coaster, one of these like drop rides, and drops them off of it, and says, look at your watch on the way down. This is a great experiment. This is the kind of thing that makes me think my experiments are boring. But it didn't work, so they still could not read the stopwatch. They described their own fall time as being longer than watching other people fall, even though gravity works, it's the same for everyone. So it must be something in the chemistry of other brain areas releasing cues to trigger memory storage. Sorry, that was a bit of a derail. So, back to particular locations that particular neurons and thus particular voxels code for in the visual field. So we can use this. And a group in Japan in 2008 did. So they showed subjects patterns that looked like this, and they submitted to the journal Neuron, which is why that's up there, very cute. But each of these patterns is a very distinct contrast pattern, so just black and white with either a pixel totally highlighted or not highlighted at all. And they would flash them in maybe checkerboard things, but the stimulation was very particular to one of these sort of 10 by 10 grid of 100 locations in the visual field. And then they measured activity from V1 and said, is fMRI good enough to actually read out what pattern someone was seeing? And they did. And these are their reconstruction results. So just based on each individual neuron encoding one sort of puzzle piece of visual space, they could then put together puzzle pieces from a bunch of different neurons and reconstruct what the pattern the person was seeing was. This is a fairly impressive demonstration, but we can do better. Part of the way we're going to do better is to try and generalize this to more complex stimuli. So the patterns that they showed, like I said, were all very black and white. They're not at all natural images. This is more of a natural image. This is something you might see out there in the wild. Well, I don't know where you hang out. So the question is, what would happen if you showed a more complex stimulus? So this is, again, a brain. I'm going to flatten it for you again. Oh, that doesn't work this one. I'm going to flatten it for you again. In slow motion this time. And I'm going to show you responses now to a slightly more complex stimulus than the last one I showed you. And by slightly, I mean massively. So this is the movie that our subjects watched in the MRI scanner. And this is a measure of what happened in the brain. I could watch this all day. I love this. This interface, by the way, was written by 2 of my lab mates who are amazing programmers and my favorite people to have around me in the lab. So you can see that, by the way, I told you that fMRI samples every 2 seconds, and you'll notice this is smoothly changing. So we're lying to you a little bit here. Anyone catch that? Watch me, I'm going to lie to you sooner or later. So these have just been downsampled. They've been smooth. So the activity linearly interpolates between one measurement and another. So it looks smooth instead of looking like a step change between things. But the changes are reasonably smooth because blood flow changes a little bit more slowly than, and by a little bit I mean a lot more slowly than neuroactivity. But you can see that there are patterns in here. There seems to be something going on. Certain areas get very excited when certain kinds of things happen. My favorite to watch is actually right up there toward the top and back here. There are a couple of places where there's a person doing something that pops up, and that area just loves it. It becomes very active in a lot of those times. There you go, people and activity. So how in the world are we going to make sense of this stimulus? Because for every frame, there's something here. Maybe there's sort of less of something here. But you need a little bit more fancy mathematics to be able to quantify what's happening in your stimulus and to be able to say whether a neuron should be active or not. So with the simpler stimuli, with the simpler stimuli, it was very straightforward to say whether a neuron should be active or not, whether there was some contrast reversing thing going on here or whether there wasn't. But now with these stimuli, you can't just code your stimulus in that way. You have to come up with a way to process your stimulus, to filter your stimulus, to determine whether a particular location should be active or not. And so instead of just using on or off at one particular location, we use a mathematical operation called convolution with patterns that look like this. So what happens when you take a little tiny pattern that's maybe 15 pixels by 15 pixels and multiply that by the luminance values in an image that's maybe 500 pixels by 500 pixels, you get out a number for every location that you put that on top of. I'm going to spare you most of this math because I went into math a lot the last talk I gave and people started glazing over a little. Suffice to say, what comes out of this operation is, it's basically a matching operation. To say that, is this part of the image, does it look like this at all? Does it have sort of an edgy thing at this particular orientation? Or does it have an edgy thing at this orientation? So now you don't just have to have, say, 10 by 10 or 7 by 7 or however many possible locations. You have that many locations times different orientations. And you have that many times different widths of the line, different sort of fineness or thickness of the line. So now we have a lot more possible filters. Instead of just a location filter that says this part is important, we have a more complex pattern that we're going to see if there are any voxels or neurons sensitive to this kind of pattern. And we have a pretty good idea they would be because of other experiments, but no one had ever tried to do it in fMRI, two moving images, with this complex of the stimulus before our lab did. So I'm going to hand wave a little bit more math, and I apologize for that. The way that we actually construct these experiments is we sit a subject in the MRI scanner, we scan their brain while they're watching about 3 hours of movies. Okay? It's a long time. In case anyone thinks it'd be fun to get their brain scanned, it's not. You are not. Remember, these are 2 millimeter bits of brain that we're measuring. That means you're not supposed to move more than 1 millimeter. Can you sit still for, sit still as in not move 2 millimeters for 2 hours? No, not fun. We get a lot of requests to be scanned, and people just, they don't know. So after these 3 hours of scanning, we then have an idea of the possible filters that the brain might be using to process this image. And this could be, one could be this location, one could be this location, one could be this location and an orientation. And so there are hundreds, thousands of possible filters that a neuron might be responsive to, or that a neuron might be implementing, or a voxel might be implementing. So the question is, which of those are important for each individual neuron? And we use some fancy math called regularized regression to determine which of these possible filters are important for a particular voxel. Okay? Moving on. What we can do, once we've determined which of these filters are important, is say, okay, now we know what this neuron cares about, and we have another stimulus coming in. We show them another 15 minutes of movies in addition to those 3 hours. And for this 15 minutes of movies, we process these with the same set of filters. And we get out some numbers. We multiply those numbers by how much each voxel cares about each of those channels. And we add them up. And what we get is a prediction of neural activity. So we get, this was meant to be a slide, but somehow got left out. What we get is, our curve looks like something like this of neural activity. And we get a prediction of neural activity that's based on all of the different things that we have hypothesized are important for that neuron's response. And the things that I'm talking about so far are just little local edges at particular orientations and particular ways. And so for each frame of the stimulus, this is time, this is stimulus frame 1. Maybe there was something really lovely oriented that way in the stimulus at that time frame. And we are trying to predict a voxel that really likes that. So it would end up having a high predicted response here. And we get some prediction that maybe matches this, maybe doesn't. But then we can try and match, or then we can come up with a number that is the correlation between our predicted response and our actual response. Yes?
So if that graph is linear, you're really doing this in 3D, is that?
So this is a time course for one measurement that we've made. Remember, we make 30,000 measurements across time?
For one voxel.
For one voxel, yes. Clear? Okay. Yes?
Was the measurement done, or did you attempt to compare a sequence with both eyes, one eye, and then the other eye?
So that is actually a variable that does affect neural responses in V1, but we did it binocularly with both eyes. Mostly because the differences in whether neurons respond to one or both eyes, neurons that respond to one eye are often right next to neurons that respond to another eye. And by right next to, I mean less than a millimeter away. And so if they're less than a millimeter away, we're not going to be able to resolve that difference with fMRI. Like I said, I don't like fMRI. It's unfortunate, it's the best we've got. Yes?
You showed a 2D pattern laid out, basically 2D on the brain. Is 3D also mapped 3D within the brain, in the voxels?
Well, there is no, so that's a good question. There are two answers. First of all, the world is 2D as far as your eyes are concerned, right? Because all you have is a retina. And so that retina is essentially an image, right? So to get depth from that is something that I'm building up to, okay? So, but the point is that the input is flat. Your retina's flat. And so you have, the only cue you have, there are a couple of, that's not true. There are about 8 or 9 cues for depth. You can get depth only by inference from separate inputs for two of your eyes, right? And if something is, you block out my face with your thumb and close one and close the other eye, and my face moves out from behind your thumb, because you get slightly different inputs. And so that gives you depth by comparing two things. But that's a computation that has to be done. You can also get depth by moving like this. And then whatever moves most quickly is closest to you, and whatever moves more slowly is further away. It's called parallax. So there are a lot of cues for depth, and there are some other ones as well. But mapping it in 3 dimensions, not only is the world really 2D according to your retina, for our purposes, the brain is pretty 2D as well. So what do this again. So, this one. So what I've shown you here is the cortex. This is the outer layer of the brain. And this is where most of the computations that we study are going on. And so what we have to do when we do an MRI scan, it is a volumetric scan, as I showed you, with that grid over the brain. And we have to determine which voxels are intersecting with this sheet in 3 dimensions and which ones are inside it, in the white matter, or outside it, or wherever. And the voxels that we're interested in are this 2-dimensional sheet of voxels. So 2D is mapped to 2D, and that's what you get. That's it. And where we get 3D out of this, and how we're able to do precise things like touch the 2 corners of our computer like that, or judge who's closer to me than another, is still a little bit mysterious. It's reasonably well understood, but there's more to be studied. Oh dear. Yes. So, going back to trying to quantify, the intent of this graph is that what we want to be able to do is quantify whether we've actually done a good job of trying to predict brain activity or not. And the only way we can really do that is to try and make a prediction of some future brain activity. Okay? Not a lot of people do this. Yes?
In the functional magnetic resonant imaging, you're talking about activity. Can you be more precise with respect to what is acting? Is it neural depolarization, for example?
So I would like to take that question later if I can, because that is a complex answer, and you're exactly right, I'm hand-waving out and glossing it. The activity measured by fMRI is blood flow. It is the presence of hemoglobin and other magnetic things in each individual cube that we can measure. And the presence of hemoglobin correlates with active areas in the same way that your skin flushes when you exercise. Your body can reroute your circulation. Your circulatory system operates at something like 1/3 or 1/5 of its total capacity. So you have a lot of room to squeeze veins closed and open them up and move blood around, particularly in your brain, which is a hugely metabolically expensive organ. So if you eat a large pizza over the course of a day, 1/16, one slice of it, goes entirely to vision, just to see. Your brain uses something like 1/4 to 1/5 of your resting metabolic state, and vision uses a fair chunk of that, about 1/4. So about 1/16 of your basal metabolic energy is just going to see. Not breathing, not running, not talking, just seeing. So it's important to be able to efficiently allocate your metabolic resources in your blood. And so it is pretty tightly correlated with things like neural depolarizations. It's actually most correlated with synaptic activity. So when there's integration of information when one neuron touches another, that's expensive for reasons I'm not going to get into right now. But please ask me later because, like I said, I love those questions. So
if you're blind, you should not eat so much pizza. Yeah.
maybe if you watch a lot of TV, you'll use a lot of energy. Who knows? Don't quote me. I am a doctor, not that kind of guy. So the question, what I'm trying to get to is, okay, now we have this idea that the brain is responding to some oriented pattern at some position. And we can make predictions of future brain activity based on that. We can get a graph for every single voxel in the brain as to what the prediction should have been, and then we can compare that to what the measurement actually was. And the correlation coefficient between those is going to range from 0 to 1, which is very nice because then it tells us from 0 to 1 how well we did. Okay? So I'm going to show you a color map now of exactly where we can predict brain activity. Okay? So this is, sure enough, the back of the brain. And it's in this area that I pointed out to you before, primary visual cortex, or V1. And to some extent, areas a little more anterior than that, a little more toward the front of the brain and the front of the brain. Remember, this is occipital pole, front right, front left. So this is not bad. This is pretty good. We have a handle on some of vision anyway. And what we can do with this is now, instead of just reconstructing one of these very simple patterns like Miyawaki and Kamitani did, we can reconstruct arbitrary patterns. And so this is a study that my laude Shinji did. Many people in my lab collaborated on it, but Shinji was the first author. That came out in October of 2011. This is the stimulus that people saw. This was the extra 15 minutes of the movie that we did not use to decide what was important in the brain responses, that we didn't use to fit our model. And this is the reconstructive activity from that. And I'm going to show you just a slightly different view of the same thing.
Wow.
So that's not bad. We put this on YouTube and we got 2 million hits. And that was more than any of us ever thought we would get. This was not my work. I should be very clear about that. This is the reason that I came to the lab, but this was not my work. So you can see that some of these are better than others. And what I'm going to try and convince you is that our job is not done. This looks really good, but this is by no means the end of the story for vision. Yes?
So this thing on the right is now again 2D. So
yes, exactly.
Where do you select that?
That is one reason that our job is not done. So the way this is done, let me play this for you one more time. The way this is done is to predict that there's going to be some brain activity in response to this image, and then to take a database of 18 million seconds of video that we downloaded, that Shinji downloaded from YouTube. It took a long time. And to choose which of those 18 million seconds would have elicited a response that was close to what we observed. Does that make sense? So we have a deck of 18 million seconds, and we're going to draw a few cards from that deck and guess, and call that our guess. Okay, so we're basing our guess on what should have elicited this response, based on what we know about the brain. So these are, exactly as you pointed out, two-dimensional images, and we are processing them in 2D with this very local computation. So even though this looks very much like the real thing, it's kind of an accident because there was a similar image out there. This model cannot describe to you that this is a face. It cannot describe to you that this woman is close to us. It has no idea about any of that. Yes?
Is that why the colors are all off?
The colors are off because this is an app. So I told you we're picking cards from a deck of 18 million seconds. These are the top 15 cards that we picked. And what this is an average, just a pixel average of all of those. So the colors are off partly because we're not factoring color into this model, because that's also very hard to measure with fMRI. Yeah, my favorite comment on the YouTube actually was someone said that you took a young black man and made him into an old white woman. It was a face in the right position. They were both recognizable as people, so that's good. Yes?
When you say summa better capture the summa line, so did you find some kind of pattern? What kind of thing can be reconstructed better than the others?
Sure. So the things that are most present in YouTube videos can be best reconstructed. Some people are very dissatisfied, by the way, with the idea of picking one from a deck of 18 million. It's 18 million. That's a lot. And people, I had a discussion with this at another talk that I gave, in which someone was saying, but you're just matching. Well, what's memory but matching, right? So we've just made a statement about what our memory is. Our memory are things that could plausibly exist in the world as sampled by YouTube. And we're picking from our memory. And we're saying this is what I remember that this was. So our memory is not perfect. 18 million is pretty big. It's better than anyone else has done. But it's not everything. So it turns out that YouTube has a lot of clips of faces.
So what you're saying is that in addition to more advanced development in your field, neuroscience, the computer database size, whatever kind of capacity also will affect this.
Oh, absolutely.
Right? Only if you can capture as much as big
yes.
database. Okay.
Yes. Yes.
Is your matching for the whole image, or is it possible to break it down and match different parts of the image?
So this is good question. Each individual voxel, remember, has only a limited little window onto the world. So each voxel basically casts a vote for an image. And what you do is you choose 2,000 voxels of our 30,000 across the whole brain that you were able to best predict, and you let each of them cast a vote. And you total up their votes, and you say it was this image. So each voxel is picking apart one part of the image, and you're combining all those together to get that this image overall was the best. So yes, in the end, it is an overall match to the image. But it also doesn't do segmentation. It doesn't say that this is an object and this is not an object. And if we have time to get to my work, that's what I'm working on right now.
It's very impressionistic.
People say that, yes. It turns out, just take some images, take 4 shots of yourself from slightly different poses, average them, and this is what you get. People love it, but that's because people don't do math with images as much as we do. So here's, actually, that's the thing I want to pause on right there. Too late. Where's my cursor? And never mind. So you notice the elephant was kind of lousy, not a lot of elephants in the database, and not any clear representation of what's a real line in this model. So remember, we're just talking about pattern matching in an image. So if there is, say, a shadow, if there is a patch of dark like this, where there's a very strong dark-light difference, any kind of computation we do on the image is going to think, oh boy, there's something important there, and there's something important here, and there's something important here, and there's something important here. And some of these are just not as important as others. Shadows are not that important. Textures, they tell you different things than the outlines or the silhouettes of objects do. So let me see if I can move on a little bit more quickly and show you something else from this that you may have missed, which is that primary visual cortex, come on. Ooh, no, that's not good. Primary visual cortex is this swath that I've drawn a line around right at the back. And it's primary visual cortex because if you measure the response to a wedge going around, you can measure that the wedge goes from here around to here. So what happens here and what happens here is you get the visual world again. You get the half of the visual world that V1 responds to re-represented. You get 2 maps instead of 1 map. And not just 2, but you get 3 or 4 or 5 or 6, depending on how many scans you do of this rather toughy stuff. So it looks like there's not just one representation of the world out there, that there's this sort of puzzle piece representation that each neuron represents a little bit. There's actually another representation of it, maybe with different kinds of puzzle pieces. and maybe with different information conveyed by those neurons and those voxels, if you can measure them with fMRI. So what this leads to, or what this clearly shows, is that vision comes in the eye, goes to the thalamus, which you mentioned earlier, goes to V1. From there, it moves on to other visual areas. So there are a plethora of visual areas, and I mentioned that if you hit your head and damage V1, you get a blind spot. If you hit your head and damage these areas, you can get some really weird stuff. You can have trouble moving your eyes to a particular spot, even if you can see it just fine. You can have trouble recognizing objects, even if you can say what they're made of. There's a famous patient who goes by her initials, which are DF, who had carbon monoxide poisoning and damaged an area of the brain that was sort of around the corner from V1. And she could describe to you, you show her something, and she says, well, it's metal. Is it, I don't know, is it a kitchen utensil of some kind? It's a flashlight. She just couldn't tell you it was a flashlight. And you tell her, okay, well, can you reach out and grab it? And she says, well, I can't really see it. Just reach out and grab it. And she will reach out, and she will size her grip exactly appropriately, and she will pinch it exactly where its center of mass is and pick it up. So vision gets way more interesting in this local image computation. And that's where it starts getting very interesting to say, well, what kind of filter could you possibly design that would capture something like that? What kind of processing of the image is going on to get there? Yes?
Well, strictly speaking, in terms of blind sight, isn't that the secondary pathway that goes through the tectopulmonar system into the cerebellum for hand-eye coordination? Is that secondary pathway? Because not all optic projections go to cortex. There's just, I said about 15 or 20% of them go off directly to the cerebellum for motor control.
15 or 20% seems high to me. I'm pretty sure it's not that high. I'm pretty sure it's like 5. And there are two pathways. That is not the primary other division in the visual system. The primary division is between the ventral pathway and the dorsal pathway. So the outputs from V1 can go in a couple of directions. They go to V2, and from V2 they go to either V3 or V4, and it gets a little hairy by that point. But the principal division of the nervous system is that some visual input is for action and reaching and grasping, which does connect to the cerebellum sooner or later.
See, that's my concern, is that, I mean, there's another famous patient like that who is able to, you put a slot in a board in her scotoma, and she can orient her hand to match the orientation of the slot to reach through, even though the slot's in her scotoma. And so I'm talking about an earlier separation of ventral versus dorsal.
That's the same patient, actually, or they've done that experiment on DF as well. And there's another patient who has a complementary lesion who injured a part of her parietal lobe and could describe what an object was just fine. They thought it's a flashlight. And you say, reach out and grab it, and she does this, feeling around on the table for exactly where it is. And so there are many different areas that are involved in many different aspects of vision. And much as I would love to talk about them all, I'm really running out of time pretty seriously. So I'm going to go with the cartoon version of this, which is that these are all arranged in this loose sort of hierarchy, where the information comes in to the eye, goes to the thalamus, goes to V1, and from there it basically gets more complicated as you go downstream. And I'm going to talk principally about, or I'm going to talk only because I'm totally out of time, about the ventral pathway and the process that your brain goes through as you're trying to recognize something. So the last stages of this ventral pathway have been studied most thoroughly, and they have been shown to be sensitive to things like faces and houses. And if you now design an experiment, much like our first simple experiment, we just have a rotating wedge. If you just have an experiment that shows images of faces, and a face, and a face, and a face, and a face, and then a house, and a house, and a house, and a house, you can find some areas of the brain that will respond very robustly. And again, this is just one voxels time course that I'm drawing for you, or that's plotted here. You can see that some voxels, for example, this voxel, responds very robustly when there's a face, and not at all when there's a house. Very robustly when there's a face, and not at all when there's a house. Exactly the opposite here in a different region of the brain. So you get almost no response to a face and very robust response to a house. So this is very interesting. The other thing that's very interesting about this area, which if you're reading the slide very closely you may have picked up, is that these areas also seem to be active when you are imagining either a face or a house. So if you have a person look at a face or a house, this is the response you get. If you have them imagine a face or a house while looking at a blank screen or having their eyes closed, you still get differentiable responses. So now we're getting somewhere. Now we can actually talk about maybe mind reading. If you can talk about the contents of someone's imagination based on your knowledge of their perceptual processes. Now it's getting fun. So this actually has been used clinically. There was a study that was done by the lab of Adrian Owens and Monty, I don't remember his first name, was the first author, in 2010, where they scanned a bunch of patients that had been in comas for N months, and they found that some small subset of these patients who had been diagnosed by their doctors as in persistent vegetative state, no response, couldn't get them to blink their eyes, to respond to snapping in their face, nothing. But when they asked these subjects, they put them in the MRI scanner and they told them this. They said, we're going to ask you a series of yes and no questions. If the answer is yes, we would like you to think about walking around your house, which activates this area toward the bottom of the brain. It's not obvious this is the bottom of the brain.
The other way around. Think of walking around your house for no.
Yes, I'm sorry, for no. And think about playing tennis for yes, which is a separate part of the brain that has to do with controlling your motor actions, which is separate from the entire visual system. So this is a very big distinction. So no is think about walking around your house, and yes is think about playing tennis. And then they would ask them, is your name Joe? Is your daughter's name Jill? Do you live in Berkeley, California? And so forth. And what they then did was scan these people's brains as they asked these and track the activity. And sure enough, there was some small fraction of the subjects, maybe I'm going to make this up right now, I'm going to lie to you and say it was about a fifth. I think it was about 6 people out of 30 or something like that, who responded 100% correct to these questions. And that's pretty striking. So this, I mean, you can see from some of the heads that some very bad things happened to these people. And they were able to communicate with them. And of the 5 or 6, there were 2 that eventually, when the doctors re-examined them with the knowledge that they had these 100% correct answers, they were then able to find a little bit of eyelid flutter or something that gave an indication that they were, someone was home. But the others, nope. Only way to get at the contents of their intentions and awareness was through something like this. So this group is now working on some other way to measure this kind of thing through some cheaper method. MRI is quite expensive. And you can't afford a $1 million magnet in your house every time you want to say, please give me some more coffee. So this is very cool. And there are obviously many possibilities to implement some of the more sophisticated designs and models of what the brain is doing, such as the one I showed you before. So I'm going to just touch very briefly on my own work. I think I have 5 minutes, so I'll take 5 minutes anyway, if you don't mind. Good, last chance.
What did you say?
So we've told you about how early stages of the visual system seem to encode information about very local patterns of whether there is an edge present in an image, in a particular window on the world. So a single neuron or a single voxel would respond maybe to an edge here and not a vertical edge here at that stage of visual processing. And then that neuron talks to another neuron, that neuron talks to another neuron, that talks to another neuron, and sooner or later, somewhere, you get some kind of coding of the semantic category of the object, that this is an animal, that it is a tiger. So it's not just faces and houses, by the way. I've left out a little bit for time. There are other areas that seem to respond to animate versus inanimate. That seems to be a big difference that your brain just fundamentally encodes, no matter where the animal occurs in the visual field. So obviously what's missing here is what in the world is happening in the middle? How do you make this transition from this very local coding of shape into these very complex semantic categories that bear much less relation to the actual stimulus as it was coming in? They're abstract quantities about the real world you want to know about, but it's very hard to get them out of an image. So the main idea that people have is that there's some sort of segmentation going on, that there's some prioritization of some of the edges, but not all of the edges. And there's a prioritization of this is a foreground object that might eat me, or this is not. So the problem with this is that it's very difficult to quantify where the edges of objects are if you're using movies such as the ones we used before. Because computer vision is not perfect yet. There are reasonably good edge detection algorithms, but they find a lot of edges that are shadows and that are textures, or they'll, if someone's wearing a vest, for example, they'll chop the arm off of someone and not assign it to the same object as the person. So these are imperfections. And it makes it very hard to say, okay, I think that this particular neuron or this particular voxel somewhere in V2, 3, or 4 is responding to silhouettes, for example. Because how do you quantify a silhouette if you're using a complex stimulus? So some people have done very simple studies where they use just simple shapes that look like Pac-Man or something. And they found that there are some neurons you can find in some areas of the brain that respond to sharp curves compared to smooth curves. But these are very, very simple stimuli, and how do you generalize that to something more complex? So this is where I come in. And I made these. You can tell they weren't done by Pixar. So the beauty of using rendered images is that these are all based on three-dimensional models of the world. So these are all graphical objects that are defined by some vertexes, and there's some mesh that connects them, and there's some surface that defines, and you can define where all that is because you have to decide how light is going to bounce off of it. This is how computer graphics works. This is how video games work. I don't know if any of you are, I think my demographic is maybe slightly underrepresented here. Sorry to everyone. But the people that actually played the sort of early 3D video games, remember that they were very polygony, they were very blocky. And that's still the way that things are done. They just, computers are faster and they can now render smoother curves with more data points on them. Point is, if you have an image like this, you can say exactly where the objects are. You can say exactly where there are contours that are associated with changes in depth between an object and the background. So if this were a real scene, you threw something, you hit the alligator in the tail here, and you would not hit it here. So this is an important edge. This one, maybe less so. This one at the edge of the alligator's mouth, maybe less so. So now, with these images, I can make a model of where the edges of objects are. And instead of computing orientation based on the pixels of the image, I can compute orientation based on the silhouettes of the objects and say that this is really the edge that is important, and see if that model does a better job of predicting brain responses than the model that I described earlier that just quantifies pixel differences. So now what I'm going to show you is where this model predicts well, and I hope you can see that this is just sort of a half step further than the other one. So I'm going to show you a little bit more explicitly by, remember this is now a correlation between my model predictions based on this idea that the brain responds to silhouettes and the actual response of the brain at every single location in the brain. So for some voxels, for some locations in the brain, this works very well, and for some, say out here in auditory cortex, doesn't work at all. But the important thing is that there's this sort of patch in here where it seems to do quite well, and there's patches back here in V1 where it doesn't seem to do very well at all, as if this is the right model for those particular brain areas. So now I'm going to show you a subtraction of the predictions between the two models, and this is the figure that makes me happy and the one that I am going to close on now, that shows that this model of just the luminance edges, the pixel changes in an image, does a good job of predicting responses in early visual cortex, and a model that's based on object edges, specifically for objects and not other things, does a better job of predicting responses in these intermediate visual areas, in LO and V4. I'm sorry, LO is the lateral occipital cortex. It's the area that was injured in the woman with carbon monoxide poisoning. We couldn't tell you it was a flashlight, possibly because she couldn't tell that the object contours were there. So that is all I have, and I'm happy to stick around for questions. Thank you for your attention.
Thank you.
So there's an interesting article in the latest Scientific American that talks about individual nerves, like the Jennifer Aniston nerve.
Yeah. Christoph Foxworth.
And that's, of course, very high resolution down to an individual nerve. And I just wonder if there's any kind of mapping or any correlation between those studies and the kind of studies that you're doing.
Well, we all want that data, basically. I mean, the best data for neuroscience is human electrophysiology, which unfortunately you can only do on people that are undergoing surgery for epilepsy. Because they actually have to have a window cut in their skull in order to resect a little part of their brain that is going haywire. And so, in order to map out this area, they often have to have an array of wires poked into their brain, which sounds horrible, but is actually not painful at all because there's no pain receptors in your brain. And people record for hours at a time in the week just before they undergo surgery. This is incredible data. This is great. So yes, it's amazing stuff and gets published in very high-profile journals. The relationship between that and the particular features of shape that I'm talking about is unclear. Partly because they handpick their stimuli not to sample sort of a wide variety of shapes, but in that particular study, they actually picked their stimuli in order to sample things that the person was really into at the time. And so they found a tabloid junkie and showed him pictures of Jennifer Aniston and Halle Berry. And sure enough, found responses in the part of your brain responsible for storing memories. Sorry. Background for other people that may not have followed that. This study was recording from single neurons in a region called the hippocampus. Anyone see Memento?
Yeah.
The hippocampus is what broke in Memento. Unlikely from a gunshot wound, it's right in the middle of your head. We forget it because it was a cool movie and a great talking point for neuroscientists. So the hippocampus turns out to be critical for storing memories and to have a lot of very specifically tuned neurons that seem to be selective for one face and not other faces. For example, Jennifer Aniston's face and not another face. So their measure of activity is a direct measure of electrical impulses discharged by neurons. And they can show that a single neuron discharges like mad when you show a picture of Jennifer Aniston, which is cool. Yes?
You had said earlier in the lecture, just really briefly, that it's really difficult to be able to do measurements like this for color perception.
Yes.
Why?
Isn't that important? The two things that people think are phenomenally important to your sense of vision that are actually two of the least important things are depth and color. Three-dimensional disparity, depth and color. So 3D movies are a humongous thing right now. Turns out regular movies, no one ever has a problem telling what's behind what. I mean, there's a certain qualitative aspect to the 3D where you can maybe reach out and touch things that brings things right in front of your nose rather than being 20 yards distant from you. But you see the world. I mean, my advisor's stereo blind, and my former advisor was actually colorblind. True story, completely true story. And how many of you have seen I Love Lucy? Right? Do you have any trouble recognizing shapes or people in that? So color is it's a minority of neurons that specifically code for it. And they tend to not be clumped. They tend to not, similar colors don't necessarily cluster together, which means that if you make one measurement in fMRI, you've got a green one, a blue one, a purple one, a yellow one, or actually it's contrast channels that are like blue, yellow, and red, green. But they're right next to each other, so you can't distinguish whether one is responding or the other because they're all locked into the same measurement.
Well, the reason why I ask is because I had heard on, I think it was on Science Lab on NPR or something, that they were talking about women who, there's a small percentage of women who have an extra
super color people
code that are super hypersensitive.
Yeah,
and that there's only a small fraction of those women with that gene who can actually perceive the extra colors.
Yeah. That's very cool stuff, but as far as I know, none of it's been done. Nothing has been followed up in functional magnetic resonance imaging based on that. It's very cool stuff. There's a lot you can do with just behavior to decide to figure out what's going on. Yes?
Is there any effect that you know of functional magnetic resonance imaging, either the electromagnetic energy that's deposited or the gradient that alters the signals you're getting?
Boy, I hope not. That's my goal.
But I mean, looking at versus embedded.
There actually was a study that came out very recently that was done in extremely high field magnets. So our magnet here is 3 Tesla, which the junkyard magnets that pick up cars are 1 Tesla. So this is a very, very strong magnet. And there are some magnets that are 7 Tesla. There's one in Minnesota that we have some collaborators that use, actually. We're trying to get some time on it. If you run around in the room with the magnet in it, then you are experiencing a very quickly changing magnetic field. And that can induce currents in your head. There's a weird thing that happens if you have to stick your head into the bore of the fMRI and move it around, where you get this metallic taste in your mouth. It's unpleasant, but it's not, it's fine. There are no known health consequences. The safest scanning technology that's ever existed. Way safer than PET or X-rays or anything else like that, because there's no high energy radiation involved. Yes?
Just following up that, there are indeed many reasons to love fMRI.
There are.
Which brings us back to your comment at the start about people who hate fMRI.
Yes.
Okay?
Yes.
And you can love fMRI, but absolutely hate the premature closure.
Yes, absolutely.
Okay? And I know you've read Ed Bull's paper.
Yeah.
And it did in fact debunk a lot of the so-called results.
Sure.
You've gotten to, okay, so
sure.
So if we continue on this, there are then there's the extension by people like Sam Harris to political and social issues, which one is really premature closure. Also, this brings us to my question. In the case of say Harris and the people that Ed Bull criticizes, we have in a lot of cases subjective states mapped to particular locations in the brain, okay? Or to put it in mathematical terms, to tensors of order 0, okay? In the kind of work you're talking about here, we have a much more sophisticated type of reduction, if you like. You're bringing in time, you're bringing in vector spaces.
Yeah.
Have you thought about this? Have you thought about, if you like, the order of tensor that we're going to need to really map what is actually occurring in people's minds?
So in map, was it occurring in vision or in these kind of social cognition domains that you're talking about?
Let's
I try not to. I'll just say I try not to think about the latter.
Yeah, let's go. No, let's go the whole hog. Let's say a tensor of order 4, such as Einstein was using in general relativity.
Well, the space of things that neurons could possibly respond to is just vast. It's humongously vast, even for vision alone. So this is, let me take a bit of a digression and come at this from something I'm slightly more familiar with, which is that you don't think about this very much, but an image that is, say, 100 pixels by 100 pixels, each of those pixels maybe could have 255 values, right? So there's 255 shades of gray encoded in each, just a black and white 100 by 100 image, which is a really small image these days. We've got like megapixel displays now, retina displays and absurd things like this. There are millions of values combinations of values that image could take on. Most of these images look like television snow. They look like black and white checkerboards of varying shades of gray. And those just aren't ever images that you see, because in the real world there are objects and light bounces off of them, and objects tend to be kind of big, and so the world looks kind of cloudy statistically, meaning that neighboring pixels in an image actually tend to be close to each other in values that they have. So there tends to be like blocks of blue and blocks of white, whatever else. So the space of all the possible images is actually not used in the real world. And so the space of all possible emotions or feelings or whatever dimension you want to measure is going to be vast. And I don't know that you could define in the abstract what space you would need to represent it all, because there are going to be subdomains of it where your feelings cluster into clouds as well. Is that an okay answer for that?
It is a very responsible answer in that it's reflecting the incredible complexity of the task we set ourselves.
Yeah. Yeah.
One more question, and then I think we're going to invite Mark to come back.
I was wondering, I know there's this experiment where they show these people a video and they say, like, count look at the basketball and look how it moves. And then, or there's a gorilla that goes by. And so people focus on like one thing when they're looking at it, and does that affect any of the stuff in the
Absolutely, great question. So the next target for serious investigation, as far as I'm concerned, is attention. And there's a lot of studies of attention that have been done. And one thing that's absolutely known is that if you, say, attend to one side of the visual field, you've got like two stimuli coming on, one's like circle, square, blah. Like find the circle on the right in two streams of shapes happening. The responses to the things on the left are humongously diminished. Definitely in fMRI and at the neural level as well. And the effects of attention on, I told you there's this hierarchy of stages. The effects of attention on V1 are small. So in other words, if there is something present in the visual field, a V1 neuron is likely going to be responding to it regardless of what you're attending to. But a V4 neuron, maybe not. Maybe its response is pretty squished. So there's, my mother will tell you this story about me, that when I was a kid, I would go looking for something and she'd say, it's on the table. And it was on the chair right next to the table. But I would look at the table and my attention was like so zoomed in that I'd be like, Mom, it's not here. But I probably, literally, did not see it. I mean, it didn't register at whatever level of processing it needed to register for it to reach my conscious awareness. So yeah, attention's a great big deal. And there's all kinds of effects that attention can have. And you can direct your attention a lot of different ways, to locations in space or to particular kinds of features, like find the red thing, find your red shirt in your laundry, right? It's pretty easy. You just go for the red thing. So stay tuned for more on that. But that's all. Thanks.
Thank you.